Biochemical test to identify food nutrients, starch. Iodine test, solutions required, food sample, water, iodine solution. Procedure, grind a small amount of food with water, add a few drops of iodine solution. Observation, purplish blue color appears. Other fact, positive result indicates the presence of starch, glucose, reducing sugar. Benedict's test. Solutions required, glucose solution, Benedict's solution. Procedure, add Benedict's solution to glucose solution, heat in a water bath. Observation, blue, green, yellow, orange, brick red precipitate. Other fact, the color change depends on the amount of glucose present. Sucrose, non-reducing sugar. Benedict's test after hydrolysis. Solutions required, sucrose solution, dilute sulfuric acid, H2SO4. Benedict's solution. Procedure, add Benedict's solution to sucrose solution and heat, no color change. Add dilute sulfuric acid and heat, add Benedict's solution again and heat. Observation, initially no color change. After acid treatment, blue, green, yellow, orange, brick red precipitate. Other fact, sucrose must be hydrolyzed into reducing sugars before Benedict's test gives a positive result. Protein, Biuret test, solutions required, protein solution, such as egg white or dal extract, sodium hydroxide, NaOH, copper sulfate, CuSO4. Procedure, add an excess amount of sodium hydroxide, add a few drops of copper sulfate solution. Observation, purple or violet color appears. Other fact, positive result indicates the presence of proteins, peptide bonds, lipids, fats and oils. Sudan. 3 test, solutions required, oil sample, such as coconut oil or gingerly oil. Sudan 3. Reagent, procedure, add Sudan 3. Reagent to the oil sample. Observation, red fat globules appear. Other fact, positive result indicates the presence of lipids, fats and oils. Quick reference, starch, purplish blue color. Glucose, brick red precipitate. Sucrose, no color change initially. Brick red precipitate after acid hydrolysis. Protein, purple or violet color. Lipids, red fat globules.